Hello everyone, welcome back. So in the lecture 1 we have discussed about briefly about the galvanic cell and the electrolytic cell. Galvanic cell and the electrolytic cell. Okay, and the electrolytic cell. Okay. So in the lecture 1 we have briefly discussed about what is galvanic cell, what is electrolytic cell. Now here we go. What are the basic components of the galvanic cell? If I say, what are the basic components of the galvanic cell? Okay, basic components of the galvanic cell. Basic components of the galvanic cell. Then, what I can say, there are the three basic components. Number one, oxidation half cell. Number one, oxidation half cell. Okay. Now the number two, reduction half cell. Reduction half cell, and the third one is solid bridge. Okay, so these are the three basic components of the galvanic cell. Okay, we have also discussed about the construction of the Daniel cell in the previous class. Okay, so this is the construction, or we can say components of the Daniel cell. Here we have concluded up to NAT reaction. So this is the NAT reaction of the Daniel cell. This is the NAT reaction for the Daniel cell. NAT reaction for Daniel cell. Okay. So this is the NAT reaction for the Daniel cell. Okay. Now, here number of electrons involved are 2. Number of electrons involved are 2. Now, the main topic is what is cell potential? What is cell potential? Look at here. What is the meaning of cell potential? Okay. Meaning of cell potential. Difference in electrode potential of two half cells. Difference in electrode potentials of the two half cells. Difference in the electrode potential of the two half cells is known to be the cell potential. Okay, so I can say difference in electrode potential. Difference in electrode potential of two half cells. Okay, and it is represented by E naught cell, sorry, E cell. E cell, we can say that cell potential. Now, what is standard cell potential? Okay, now look at here. Okay, what is standard cell potential? If we calculate the cell potential, or if we apply the standard conditions while calculation, okay, so measurement of cell potential in the standard conditions. Now, what are the standard conditions? Look at here. Standard conditions. Standard conditions. Okay. If I say pressure 1 bar, temperature to be 25 degrees Celsius, and concentration to be 1. So these are the standard conditions. This is capital M. Okay. So when the measurement of cell potential in the standard conditions is known to be the standard cell potential, and it can be represented by E naught cell. So this is the standard cell potential. Okay. Now, SOP, now how we can measure E cell? Okay, with the help of oxidation potential and reduction potential. So you have to remember that cell potential is the sum of oxidation potential, sum of oxidation potential of anode oxidation potential of anode and reduction potential of cathode. Sum of oxidation potential of anode and reduction potential of cathode and reduction potential of cathode. Okay, so this is the E cell. Okay, this is E cell means what? Cell potential. Okay, now what is E naught cell? What is E naught cell? Look at here. E naught cell is standard cell potential and it can be equals to SOP. That means standard oxidation potential. Standard oxidation potential. Okay, and SRP is what? Standard reduction potential. Standard reduction potential. Okay, so this is E naught cell. Okay, so this is all about the cell potential and the standard cell potential. Okay, now, now this is the important topic, cell representation. Now, if I have to separate out anode and cathode, okay, here is anode and here is cathode, and in between them, one salt bridge is present, and this salt bridge can be represented by the two vertical lines. These two vertical lines. These two vertical lines represent what? Salt bridge. Okay, two vertical lines represents what? Salt bridge. Okay, so whenever you have to represent salt bridge, you just have to draw two vertical lines between anode and the cathode. Okay, now let's understand the second concept. If components are in different phases, components are in different phases. Okay, what does it mean? Let's understand. If components are in the different phases, like copper solid and U plus 2 aqueous. So you can see that this is solid and this is aqueous. So that means components are in the different phase. So how we can separate it? Okay, how we can separate it out? Okay, we just have to draw a single vertical line. So single vertical line is used to separate two components having the different phases. Okay, now the third one. If components are in same phase, if components are in same phase, if components are in same phase, then what we have to look at the example Fe plus 2 and Fe plus 2. These both are in aqueous. Okay, then we just have to use a single comma between them. Okay, so salt bridge can be represented by two vertical lines. If components are in different phase, we just have to put one single vertical line in between them. And if components are in same phase, we have to use the comma between them. Now the fourth point. Look at the fourth point. Along with the components, we can write down concentration and pressure or pressure. Okay. So along with the components, we can write down what? Concentration, or we can say pressure. If components are in gaseous phase, we have to use pressure as well. Okay. So this is the cell representation for the Danube cell. Look at this example. So this is the cell representation for Daniel cell. This is the cell representation for Daniel cell. Okay. Now, here anode is in the left side. You can see here, anode is in the left side and cathode is in the right side. Okay. Now, here oxidation takes place. Here oxidation takes place. And here reduction takes place. Okay. Now, you can see here that there is a two vertical lines. So these two vertical lines represent what? Solid bridge. This single vertical line shows that the components are in different phase, like solid and aqueous. Here components are also in different phase. That's why single vertical line has been used here. Okay, so this is the representation for the Daniel cell. Now look at the electron flow as well. Now electron flows from anode to cathode. Now the question is that what is the direction of the current? Okay, so look at it. If I say electrons flow from cathode to anode, if I say electrons flow from anode to cathode, electrons are flowing from anode to cathode. Okay, so always remember current flows opposite in the direction of the electrons. So what is the direction for the flow of current is from cathode to anode. Here I is what current. Okay, so direction of flow of electrons is from anode to cathode, and what is the direction of flow of current is from cathode to anode. Okay, there are many examples for the cell representation. We shall discuss in the numerical section as well. There are many more formulas, many more tricks, okay, about the E cell, E naught cell, SRP and SOP. We shall discuss in the numerical section. Numerical session. Okay. If you are new at this platform or new at this channel, kindly subscribe, share, and like this video. Hope you understand the topics. Okay. Thank you so much, Deepak Goyal.